you think about the world as bacteria, viruses, plant life, human life, and maybe you could have some like more nuanced characteristics in there, but when it comes to phage, we never learned about it in school, and it is like a very weird construction. It's got like a tube on the top with this geometric figure on the top of it, and it grabs a little bit of its own DNA and injects it into bacteria, and then makes the bacteria mass produce the DNA for the phage, and then it explodes the bacteria, and then that phage goes all over the place. And there are way, way, way more phages than anything else in the world. But the thing that was interesting to me is that we're not really quite sure, are they alive or not, which is an interesting question.
Well, it's the same with any real virus. Like, it depends where you draw the line of where they are, where things are alive or not. So I call them biological entities. My boss thinks viruses are alive, and that's not like, oh, he's crazy and thinks viruses are alive. It's sort of like where he draws that very fine line between this is living material and this is not living material. He's like, well, viruses, they reproduce, they reproduce with selection, they have their own genetic material. The only thing they can't do is survive without their host, and that's sort of, and they also don't really have metabolism. Well, some of them do have metabolism. So it's when you start looking at biology at this level, it becomes incredibly clear just how fuzzy our definitions of what is and what isn't alive actually is. Like, I could wake up tomorrow and be like, no, they're dead, and I could wake up the next week and be like, no, maybe they're alive. And there's no clear or fast answer here about what biological life actually means.
Yeah, it kind of strikes me as the weird, like, the closer you zoom into something, the more that you know it, the less you're convinced that you know what something as simple as alive or dead is, on or off, 0 or 1, because as it turns out, not all of the features of our beings around us follow the same rules that we do. Definitely.
It's there's compartments of different things that you like. We said there's these 6 or 7 things you need to be considered alive, and you actually, when you look at it, it's like, well, it's just a smear across like the planet. My favorite other thing to talk about when we have these discussions is mitochondria. So in America, mitochondria are taught as the powerhouse of the cell, which I've always found funny. When you mention mitochondria to any American, like, oh yeah, the powerhouse of the cell. It's like, it's this weird like American Canadian thing. You guys, they don't have that in Europe or the UK. That's not a thing. But so mitochondria are the powerhouse of the cell. They make energy for us in pretty much every single eukaryotic cell, like you, me, plants, everything that you can see. There's a mitochondria making energy. These, about 2 billion years ago, were free-living bacteria. At some point, another bacteria came and gobbled one of these up, and they decided to live in symbiosis as opposed to like eating it. And that led to like 2 billion years later to having enough energy to make bigger and bigger cells. But the thing that's mind-blowing to me is the fact that this was once a free-living bacteria, alive as anything else on the planet. Like, it was alive. It was a living organism that had been alive for 2 billion years through evolutionary history, and then it got eaten and it didn't die. But over 2 billion years since then, it's evolved to be dead. If you take a mitochondria out of a cell, it's dead. It's not a living material. It can't survive by itself anymore. It's no more alive than a virus. But that's happened through evolution. Evolution has selected for something to be dead. It's just like how you're like,
and like you think about like what other organism grabs another organism and makes it not just like work for me as a slave, but like actually incorporate it in and becomes a part of the way that your own DNA writes out and produces more mitochondria.
Yeah. Well, this is a really cool thing. There's whose book is it? It's maybe Nick Lane. He talks about how he didn't discover this; he just popularized it. But part of our genome has bits of the mitochondria DNA in it, and obviously the mitochondria also has its own genome. It has a little bit of bacterial DNA still in every cell. It has a little circular chromosome. That's how we know it was a bacteria, because it has a little circular chromosome. It has the same 16S kind of like bacteria, like a very specific gene. But it shared some of its genes with us, but not all of them. So it looks like at any minute it's ready just to pack up and go home. Like it's got its own genome. I'm going to leave you. I don't need you. It's this little thing packaged away. So it's I could talk about this for days, but it's an amazing insight into just how powerful natural selection is to see. Like, okay, it's put this gene here and that gene stayed here. There's a reason why some of the genes are in our chromosome and some of them are still in the mitochondria. and it's all due to natural selection.
So as you're studying phage research, what drew you to that? I mean, people have never even heard of it.
Yeah, I have wanted to work on phage since I was about 7 years old, I think it was. I remember the moment my mom picked me up from after school. She worked. I was at an after school club, and I asked her, because she's a microbiologist. I asked her, why don't they use viruses to kill bacteria? Like, as you know, this little 7-year-old kid who had no idea what I'm talking about. And she's like, well, they do. They're called phage. And then she sort of explained a little bit about phage to me, and she bought me some book about animal life, like the mechanisms of animal life. Like, so it was like they took everyday animals and like made them into like machines, and then you could open that, pop-up book kind of thing. They open up and you'd see inside the animal, and they had at the back was a couple things on viruses, phage, and bacteria. And I just fell in love with this idea of like these things that are the enemies or things that make you sick are out there. They're somewhere in nature. And then, I went away to university, 10 years later, I studied all those things. I studied bacterial communication for my PhD. And eventually I got to the point where I was like 2 months away from finishing my PhD. And I was on Twitter, PhD student, have loads of time to just hang out on Twitter. And someone had posted a job to work on phage therapy research in Montpellier in France. On the organism I was working on during my PhD. I was like, oh, cool. I love phage. I've had these ideas. I'll email the guy. And like, at to that point, I was like, I hadn't seen very many people working on phage, like out and about, because it still is quite a small field. It's only a handful of researchers who have the money to work on it. And most people are working on it are either doing, like I am now, a mixture of evolutionary studies and phage therapy. And there isn't a huge amount of people doing other things on it.
What do you think is possible with phage research? Phage therapy?
Yeah, that's a great question, right? It's stopgap's not the right word, but it's going to pick up the slack from antimicrobial resistance. Like, I'm not one of these people who think like phage are the solution to everything. We have the solution to everything. It's antibiotics. They are amazing drugs. They are so good. They work so well, and we've kind of squandered them and used them inappropriately. I think we're going to use phage to basically pick up the pieces where we've made mistakes. Phage has limited uses. Like, there are cases. I know I've met people now, and I'm so fortunate. We can talk about this later if you want. I've met people who have been given the phage I work on in the lab, and it saved their lives. That's amazing to me. But we should have never got to that position. We should never be in the position where it's like we have to use something experimental because we've been so irresponsible with the magic bullets we have of antibiotics.
And when you're talking about